Hello, my name is Javier Carmona, and I'm going to be presenting today to you on AMPA receptors, the molecular gatekeepers of fear, memory, and therapeutic targets. Imagine this scenario. A college student, Alex, is walking down a hallway late at night when suddenly a large spider scuttles across the floor. And instantly, Alex's heart rate spikes, muscles tense, and he feels an overwhelming urge to back away. This immediate fear response isn't just psychological, it's rooted in biochemical changes happening in his brain. Inside Alex's amygdala and hippocampus, tiny molecular machines called AMPA receptors are playing a crucial role. They're helping neurons tune in to this frightening stimulus, strengthening the synaptic connections that encode the spider as a threat. Over time, repeated encounters or even memory recall of that spider can reinforce these fear-related pathways. Conversely, learning that the spider is harmless or undergoing therapy can shift AMPA receptor dynamics to gradually diminish that fear. Today, we'll explore how AMPA receptors work, where they reside, the nuances in their structure, and how understanding them at a molecular level can inspire treatment for anxiety disorders, epilepsy, and more. AMPA receptors are found primarily in the central nervous system. Embedded in the postsynaptic membranes of excitatory synapses, they're abundant in key brain regions involved in learning, memory, and emotion, like the hippocampus, crucial for forming new memories, and the amygdala, central to processing fear and other emotions. In Alex's fearful encounter, the AMPA receptors in his amygdala are helping neurons respond rapidly to signals that say this situation might be dangerous. AMPA receptors belong to the ionotropic glutamate receptor family, which includes AMPA, NMDA, and kainate receptors. Being ligand-gated ion channels, when the neuron transmitter glutamate is released into the synaptic cleft, it binds to these ionotropic receptors, causing their ion channels to open. AMPA receptors are primarily permeable to sodium ions. As Na+ flows into the postsynaptic neuron, the cell depolarizes, creating an excitatory postsynaptic potential. This quick, millisecond timescale response sets AMPA receptors apart as fast excitatory receptors. Repeated or particularly strong activation can strengthen the synapse, a process known as long-term potentiation. In fear circuits, such strengthening could encode and reinforce fearful associations. Conversely, modification in AMPA receptor number, composition, or subunit types during extinction learning can help weaken those once potent fear memories. To understand how AMPA receptors operate, let's examine their structure. Each AMPA receptor is a tetramer, composed of 4 subunits. These subunits can be the variations of GLUA1, GLUA2, GLUA3, and GLUA4. The combination of subunits influence properties like ion permeability and channel-gated kinetics. For example, AMPA receptors can also be permeable to other cations than just sodium. The AMPA receptor's permeability to calcium and other cations such as potassium is governed by the GLUA2 subunit. If an AMPA receptor lacks a GLUA2 subunit, then it will be permeable to sodium, potassium, and calcium. The presence of a GLUA2 subunit will always render the channel impermeable to calcium. This change is principally driven by RNA editing that alters the uncharged amino acid glutamine to the positively charged arginine in the receptor's ion channel. The positively charged amino acid at the critical point makes it energetically unfavorable for calcium to enter the cell through the pore. Almost all GLUA2 subunits in the brain are RNA edited at this site, which is why calcium impermeable AMPA receptors are predominant. The most common AMPA receptor composition that is studied in relation to the brain are GLUA1/GLUA2. They are heterotromers. The ribbon diagram shows a beautiful arrangement of alpha helices and beta sheets that form the receptor's overall shape. This well-orchestrated folding is what allows the receptor to undergo subtle shape changes when glutamate binds, flipping it from a resting to an active state. At the quaternary structure level, 4 subunits assemble into a functional receptor. Imagine 4 proteins arranged like petals of a flower around a central channel. Each subunit contributes to the pore's linings and to binding sites for glutamate. This symmetrical architecture ensures that the receptor can respond robustly to even small amounts of neurotransmitters. If you swap a GLUA2 subunit for a GLUA1, you might affect calcium permeability or channel conductance. It should also be noticed that defects in the GLUA2 subunit have been linked to neurodevelopmental disorders such as epilepsy, seizures, and intellectual disability. Such variations fine-tune the neuron's excitability. For someone like Alex, subtle changes in AMPA receptor composition in the amygdala could mean a stronger or weaker fear response. Each AMPA receptor subunit has 3 major domains: the amino terminal domain, ATD, ligand binding domain, LBD, and transmembrane domain, TMD. ATD is located on the extracellular side, involved in subunit assembly and receptor trafficking. It can also influence receptor function by interacting with auxiliary proteins and modulators. LBD is also extracellular. This is where glutamate docks. The LBD acts like a clamshell. When glutamate binds, the two lobes of the LBD move slightly, transmitting a mechanical force to the transmembrane domain. TMD spans the lipid bilayer and forms the ion-conducting pore. The TMD's alpha helices line the channel, pivoting and tilting as the receptors transitions between closed and open states. These domains are carefully engineered at the molecular level so that a single binding event, glutamate binding, can ultimately translate into a large-scale event, ion flow, and thus neuronal signal. ATD, which is a mixture of alpha helices and beta sheets, provide a stable scaffold for inter-subunit contacts. The ribbon structure of GLUA1 in ATD is shown in image B, where the secondary structures are labeled sequentially, alpha helices in yellow, beta strands in blue, and loops in green. LBD, containing a mix of alpha helices and beta strands, form two lobes that create glutamate binding pockets, shown here as the red pentagon in image A, for the full-length tetrameric GLUA2. The precise arrangement of these elements is crucial for high-affinity ligand binding. And for TMD, it is primarily alpha helical segments embedded in the membrane. The hydrophobic regions of these helices interact with the lipid environment, while more polar residues face the channel's interior guiding ion flow. If we change just one amino acid in a key helix, it could alter the receptor's gating properties, affecting how easily it opens or closes, ultimately influencing how neurons respond to fearful stimuli, among other effects. Understanding AMPA receptor structure and function is not just academic, it also guides drug design. One clinically relevant example is perampanel, an FDA-approved drug used to treat epilepsy. Perampanel is a non-competitive antagonist of AMPA receptors. Unlike competitive antagonists that compete directly with glutamate at the LBD, the perampanel binds to a distinct allosteric site on the receptor. By binding in the TMD of the receptor complex, perampanel stabilizes a conformation that reduces the likelihood of the channel opening, even when glutamate is bound. This leads to decreased excitatory synaptic transmissions, helping to calm the hyperactive neuronal firing seen in seizures. In principle, drugs like perampanel also have potential implications for fear and anxiety research. By dampening AMPA receptor-mediated excitatory signals, they could reduce the reinforcement of fear memories, though this is more of a research frontier than a clinical standard. Perampanel's action illustrate how drug developers capitalize on our detailed structure knowledge of AMPA receptors. By knowing exactly where and how to bind, they can modulate receptor activity with high specificity, minimizing side effects, although perampanel does also have side effects. So before we wrap up, let's look at a specific study that highlights perampanel's potential beyond its use in epilepsy. A 2020 study by Bektas and colleagues investigated the anxiolytic, anxiety-reducing effects of perampanel in mice. Since perampanel is a non-competitive AMPA receptor antagonist, the researchers hypothesized that by reducing AMPA receptor-mediated excitatory signaling, it could influence anxiety-related behavior. In their experiment, mice were treated with low doses of perampanel, 0.25 to 1 mg per kilogram, and they showed a reduced anxiety-like behavior in tests such as the elevated plus maze and the whole board test without becoming sedated. The drug's effectiveness was comparable to that of diazepam, a well-known anxiety medication, suggesting that blocking AMPA receptors had a meaningful anxiolytic effect. Interestingly, when the researchers used antagonists to block GABA A and A2 adrenergic receptors, some of perampanel's anxiolytic effects were diminished, indicating a complex interplay involving not just glutamate, but also GABAergic and noradrenergic systems. This study suggests that at least in animal models, dampening excitatory signaling through AMPA receptor antagonism might help reduce anxiety. It's an exciting avenue for future research, potentially contributing to new strategies for managing anxiety disorders and conditions involving excessive fear, further supporting the idea that understanding AMPA receptor structure and function isn't just academically fascinating, but may ultimately improve mental health outcomes. Returning to Alex's story. The intense fear response initiated by the spider was at the molecular level partly due to the AMPA-mediated synaptic strengthening. With repeated exposure and time, changes in AMPA receptor composition or the influence of certain drugs could reduce these heightened responses. The synapses might incorporate more GLUA2 containing AMPA receptors, making them less permeable to calcium, or internalize some AMPA receptors altogether, weakening the once potent fear memory. This adaptability highlights the plastic nature of neural circuits. The very same molecular principles that create strong fear memories can also be harnessed to alleviate them, paving the way for targeted therapies. In summary, AMPA receptors are integral membrane proteins that convert chemical signals, glutamate binding, into electrical responses. Found at excitatory synapses throughout the brain, these tetrameric receptor structures from their domain organization to their secondary and quaternary architecture, underlies their crucial role in synaptic plasticity. By influencing ion flow, AMPA receptors help encode and modulate fear memories. Drugs like perampanel, a non-competitive AMPA receptor antagonist, show us how precise molecular knowledge can lead to effective treatment for neurological disorders. As we continue to unravel these molecular details, we open the doors to new therapeutic strategies that could help people like Alex overcome their fears and help patients worldwide find relief from epilepsy and other excitatory dysfunctions. Thank you so much for watching, and here are my references and resources for more information.